There are over 5,000 different minerals, some of which look incredibly similar in rocks. I mean, clinopyroxenes and orthopyroxenes are often only different by a slightly greener tinge in clinopyroxenes. So how do we tell these minerals apart then? In mineralogy, we do this by creating a thin section of a mineral and viewing it under a petrographic microscope. In this video, we will explain the optical properties of minerals and how to identify them in thin sections under the microscope. This is a petrographic microscope, and there are two main modes we use to determine the properties of minerals. Most properties are found using orthoscopic mode. However, when finding the interference figure, we insert the Bertrand and condensing lens, as well as used cross-polarized light. We'll get into that later. As seen in this diagram, there are two polarizers in the petrographic microscope. We can use these to change the vibration directions of light entering our specimen and the ocular viewing lens. Normally, in white or unpolarized light, the ray vibrates in all directions perpendicular to the direction of travel. However, a polarizer, such as those in the microscope, only allow a specific direction of vibration to pass through, meaning light emerging from the polarizer, known as plane polarized light, or PPL, only has a single vibration direction. When plane polarized light travels through another polarizer, if that polarizer has the same preferred vibration direction, it all passes through. But if the second polarizer's preferred vibration direction is perpendicular to the first, none of that light is let through. And if the second is partially rotated, only part of the light gets through. The key takeaway from this is if two polarizers are perpendicular, no light comes through the second. In the petrographic microscope, the bottom polarizer vibrates east to west and is always inserted, while the top polarizer, known as the analyzer, can be removed to produce PPL or inserted to produce cross-polarized light, also known as XPL. Most petrographic microscopes will have 4 objective lenses and 1 ocular lens. To get the total magnification, multiply the objective lens, which could be 2 times, 4 times, 10 times, 40 times, by the ocular lens, which here is 10 times. That's pretty standard. Let's start with the mineral properties in plane polarized light. Make sure you're using orthoscopic as well. There are 2 main types of sample preparations. There's the grain mount, where individual grains are sealed within a mounting oil, and the thin section, where a slice of rock is epoxied into the slide and ground down to a thickness of 30 micrometers. The first property in plane polarized light is opacity. Opaque minerals, such as oxides or sulfides, like pyrite, don't transmit any light at this thickness, and always appear black, which means we can't find their properties with transmitted light, and need to study with reflected light instead. We won't be focusing on these in this video. Non-opaque minerals do transmit light, so we are able to find their properties. Non-opaque minerals include silicates, such as quartz, and carbonates, such as calcite. Next, note the shape of the grains. How clear are the crystal faces? They can range from euhedral to subhedral to anhedral. Are they equant, with similar dimensions in all directions, like this garnet? If they aren't equal, the shapes can vary based on how the crystal is cut in a thin section, such as a cross section that shows all the cleavages, or a longitudinal section that doesn't. These shapes can be described as elongated, fibrous, prismatic, blocky, and basically a bunch of others as well. Different minerals can also have different refractive indexes, which depend on the velocity of light as it travels through the mineral, as the refractive index equals the velocity of light in a vacuum divided by the velocity of the light in the medium. The relief of the crystal is the difference of the mineral's refractive index in comparison to that of the medium. The relief can be described as low, moderate, or high, with high having the most difference, as you can see here. To determine whether the mineral's refractive index is larger or smaller than the medium's, we can perform the Becke line test. To perform this test, zoom in on the border of a grain or a crystal. I tend to use the 40x lens for this. Then focus outwards by turning the fine focus knob towards you. If the Becke line moves in like this, the refractive index of the crystal is greater than the medium, so we say the crystal has positive relief. Let's see that once more. But if the Becke line moves out, like this, the refractive index of the medium is greater, so we say the crystal has negative relief. Let's see that one again. Some minerals also have cleavage. Minerals can have a single cleavage, such as this biotite, or multiple cleavages, like this hornblende. To make sure you find all the cleavages, examine multiple crystals or grains to find a cross-section that shows all the cleavages. If a mineral has multiple cleavages, make sure to note the number of cleavages and the angles between them. For example, that hornblende has 2 cleavages at 56 degrees and 124 degrees. Minerals can form in 7 different crystal systems, and depending on which crystal system a mineral forms in, it can have different properties. The crystal system of a mineral is dependent upon the organization of atoms to form a unit cell, which resembles one of these 7 shapes. Minerals in the cubic, or isometric system, have the same composition and dimensions along all 3 axes. This means no matter how the grain is oriented, light travels through at the exact same velocity. These minerals are known as isotropic minerals. The other 6 crystal systems are not identical in all directions, meaning light travels through at different velocities depending on the orientation of the crystal. These minerals are known as anisotropic minerals. When light travels through an anisotropic mineral, it gets split into 2 rays, depending on the preferred vibration directions of the mineral, which depends on the composition and structure. In plane polarized light, some anisotropic minerals show a property known as Pleochroism. That is, as you rotate the stage, and therefore the mineral, the color of a crystal may change. For example, this mineral, biotite, is pleochroic. When the cleavage is oriented east to west, the mineral is a dark brown color. But when I rotate the stage so the cleavage is oriented north to south, the mineral is a much lighter brown. The differences in the absorption color of pleochroic minerals is due to the differential absorption of different wavelengths of light. It's also important to note, not all anisotropic minerals exhibit pleochroism, but no isotropic minerals do. So if you observe pleochroism in a crystal, you can be sure it's not an isotropic mineral like garnet. But if you don't observe pleochroism, you can't rule out anisotropic minerals. Plane polarized light can help identify a mineral, but more techniques are required to assist in the identification. So next we'll look at the optical properties of minerals under cross-polarized light. First, make sure to subscribe, like, comment, and share. Then click on this video here to continue with the optical properties of minerals in cross-polarized light.